Okay, let's unpack this. We are diving into, I mean, it's one of the most frustrating puzzles in cancer treatment.
Oh, absolutely.
You see this initial, just glorious success with cutting-edge immunotherapy, and then a swift, devastating relapse.
It really is the ultimate betrayal. The tumor just melts away, and then it comes roaring back.
So today's Deep Dives is focused on squamous cell carcinomas, or SCCs, and we're going to dissect the molecular sabotage that lets that happen.
Exactly. How these really resilient cells not only survive the drugs, but actively turn the immune system against itself.
And we're talking about a really powerful immunotherapy combo here. It's anti-PD-L1,
which takes the brakes off the T cells.
And a CD40 agonist, which kind of revs them up. It's supposed to be a 1-2 punch.
It is, but sometimes, as you said, the relapse happens anyway, and to understand why, we need to meet our cast of characters.
Okay.
On the tumor side, you have the core problem. The tumor-initiating stem cells, we can call them tSCs.
These aren't just any cancer cells.
No, they're the really aggressive subset. And the ones causing trouble here are defined by high expression of a transcription factor called SOX2. So we'll call them the SOX2 high tSCs.
So they're the architects of the resistance.
Precisely. And the other player is an immune cell, a type of white blood cell called the tumor-associated neutrophil, or TAN.
Right, neutrophils. They're supposed to be the immune system's first responders, the good guys.
They are. But when they get into the tumor microenvironment, the TME, they often become immunosuppressive. They basically become traitors.
So our mission for this deep dive is to explain step by step how those SOX2 high tSCs force the neutrophils to stay traitors.
They don't just survive an attack. They actively program the immune cells right next to them to build a kind of molecular shield. It's what ensures the relapse.
That sets the stage perfectly for what researchers call the neutrophil paradox. So where do we start? How did they figure this out?
Well, they used a very rigorous model.
Yeah.
So instead of just grafting a tumor onto a mouse, which, creates a predictable kind of homogeneous lump.
Right, not very realistic.
Not at all. They used a chemical model, DMBA plus TPA, to induce spontaneous tumors. This creates cancers that are much messier, more varied, and they better mimic the complexity we actually see in human patients.
And with that complexity, they must have seen that the TANs weren't all the same.
Not even close. Using single-cell RNA sequencing, so looking at the genetic profile of thousands of individual cells, they found 4 distinct clusters.
Okay, so what did they look like?
Well, you had clusters T1 and T2, more mature cells. T1 was generally pro-tumor, but the one that really stood out as uniquely bad was the T4 cluster.
And what made T4 so bad?
It was immature, highly suppressive, and just loaded with a gene called ARG1.
Arginase 1, that's the one famous for starving T cells, right? It just gobbles up all the arginine they need to function.
Exactly. It makes them completely ineffective. T4 is the worst of the worst.
So here goes the paradox. The researchers give this potent immunotherapy.
And the tumor shrinks. T-cell activity spikes. Everything looks great. Success.
But then they did the crucial test. They used these special mice that let them basically delete the neutrophils during the treatment.
Right. They could chemically deplete them.
Wait a second. If the neutrophils are suppressive, then getting rid of them? That should help, shouldn't it?
That's what logic would dictate. But no, it was the complete opposite.
What happened?
When they depleted the neutrophils, the tumor clearance was abolished. It just stopped, and the T cells went silent.
Wow.
The insight here is massive. These neutrophils, which start out as villains, somehow become essential for the therapy to work. They have to switch sides.
A complete personality change.
Yeah.
An immune conversion. But how do they know it's a conversion?
Yeah.
Maybe the old bad cells just die off and new good ones rush in.
That's the critical question they had to answer. And they used this brilliant lineage tracing technology to prove it.
How does that work?
They used mice where they could genetically label the pre-existing suppressive TANs with a fluorescent color before treatment ever started.
So they're marked.
They're marked. And when they checked after the immunotherapy, they confirmed those same labeled cells, the old suppressors, were the ones that had converted into the anti-tumor helpers.
So it's the same cells, they just learned a new trick. That is the definition of plasticity. So what's the remote control that flips the switch?
Interferon signaling.
Yeah.
Post-treatment, the T1 and T2 clusters, they shed their suppressive markers. They start aggressively upregulating anti-tumor genes, things like IL-12A and TNF.
And they start showing new things on their surface.
Yes, markers like LY6E And critically, MHC class II, which is what they need to present antigens and talk to the immune system, they become bona fide anti-cancer helpers.
And the proof that interferon, or IFN, is the remote control.
If they genetically knocked out the interferon receptor specifically in the neutrophils, that phenotype switch was completely blocked. The whole immunotherapy failed.
So IFN is the non-negotiable signal. It's what turns the black hats back into white hats.
It is.
Okay, let's just quickly consolidate that first part for everyone listening. We've gone from a paradox to an absolute necessity.
Right. The key finding is that the immunotherapy only works because of T-cell activity, but that T-cell activity relies entirely on reprogramming the neutrophils that are already there.
And that reprogramming, that conversion, is mediated entirely by interferon signaling. It's what gets them to display MHC class II and help out.
Exactly. But remember that T4 cluster, the one high in arginase 1?
The one that looked uniquely resistant. That cluster holds the key to why tumors relapse. We had to figure out why those cells refuse to change.
And that brings us to part 2, the spatial resistance. So the researchers asked, is this resistance happening everywhere, or are certain locations acting like protective shields?
So where matters.
Where is everything. They use something called GeoX spatial transcriptomics. You can think of it like molecular GPS to map the immune response layer by layer across the tumor.
And I'm guessing the landscape was not uniform.
Not even close. If you looked at neutrophils deep in the stroma, the connective tissue, they had successfully reprogrammed. They showed high levels of IFN-stimulated genes like GBP2.
So the remote control was working there.
It was. But the moment you looked at the tumor-stroma interface, that critical border zone, the dividing line.
The fortress wall.
The fortress wall, exactly. The neutrophils there showed strikingly downregulated IFN responses. They were still suppressive. That interface is the protective micro-niche.
And I'm going to guess who's living right at that fortress wall.
The cancer masterminds themselves, the SOX2 haughty tSCs. They accumulate right there.
Which strongly suggests SOX2 is the architect building the wall and running this whole sabotage operation.
It does. But they had to prove it. I mean, SOX2 is a known factor for stemness, but how do you prove it's the specific culprit here?
Right, and not one of the other dozen factors.
So they systematically tested the transcription factors that were enriched in those tSCs. The result was indisputable.
It was SOX2.
Only SOX2 overexpression successfully blocked the TANs from activating MHC class II. It kept them in that low-function suppressive state. The SOX2 high cells were actively putting a muzzle on the neutrophils next to them.
So what does this muzzle do? Does it just block the T-cell signals, or does it actually stop the neutrophil from even listening to the interferon in the first place?
It blunts the listening. Neutrophils they isolated from SOX2 high tumors resisted interferons both in vivo and even in vitro.
So even in a petri dish?
Even in a dish. You could soak them in high doses of interferons, and their ability to transcribe those anti-tumor genes was just dampened. The SOX2 cells create a local field of resistance that makes the neutrophils deaf to the commands of the immune system.
And the ultimate consequence is T-cell exclusion. If the neutrophils can't display MHC2, they can't present antigens, and the T-cells have no reason to interact with them.
Exactly. The research showed significantly fewer interactions between CD4 plus T cells and neutrophils near those SOX2 high regions. The T cells literally couldn't get close enough to fight the stem cells.
And the clinical validation here, that's crucial.
It is. They used what are called SOX2 conditional knockout mice. When they chemically deleted SOX2 just in the tumor stem cells, every single infiltrating neutrophil became IFN responsive.
So the resistant T4 cluster.
It vanished entirely. And critically, ablating SOX2 completely abolished tumor relapse after immunotherapy. That whole protective niche was dependent on SOX2's control.
And this isn't just some weird mouse biology, right?
Not at all. The human relevance is stark. Human head and neck and skin SCC samples showed the exact same pattern. TANs near SOX2 high cancer cells had reduced HLA-DR expression, that's the human version of MHC class II, and fewer T cells nearby.
The stem cells are running the same sabotage program in us?
So now we know who the architect is, SOX2, and where the fortress is, the interface.
So now we need to find the weapon, the specific molecular signal it's using to maintain that force field.
Which brings us to part 3, the AAPGE2 axis.
So the SOX2 architect is turning on some secret factory inside the cancer cell to make a signal. What do they find?
It led them down a fascinating path of lipid metabolism. Bulk RNA sequencing of the SOX2 high cells revealed a massive upregulation of an enzyme called FADS1.
Fatty acid desaturase 1.
Right. And using a more detailed sequencing method, they confirmed that SOX2 directly binds to the promoter region of the FADS1 gene.
Okay, so that's definitive. SOX2 is the master programmer, and FADS1 is the specific factory switch it flips on.
And FADS1 is the first step in creating the saboteur payload. It catalyzes the production of polyunsaturated fatty acids, and specifically, it generates arachidonic acid, AA.
Which is a very common fatty acid, but I'm guessing it was showing up in weird places.
It was significantly enriched in the liquid surrounding the tumor cells, the tumor interstitial fluid, but only in the SOX2 high tumors.
So AA is the delivery mechanism. But is AA alone enough to break the immune response?
They proved it was the immediate signal. If they just injected arachidonic acid directly into a SOX2 low tumor one that was previously easy to treat, that was enough to blunt the IFN responsive capacity of the TANs.
So AA is the necessary chemical intermediate. But you said payload, which makes me think it's not the final poison.
You're right. This is where the elegance of the mechanism really shows. AA has to be converted by the receiving neutrophil. It's a trap.
How does that work?
AA is metabolized by enzymes called cyclooxygenases, or COX enzymes, into something called prostaglandins. And after testing different metabolites, they identified prostaglandin E2, or PGE2, as the critical downstream saboteur.
PGE2. That is the final bullet. And how does it physically stop the neutrophil from switching sides?
It's essentially cutting the wires of the remote control. It disrupts the interferon signaling pathway by reducing the phosphorylation of key signaling molecules, STAT1, JAK1, and JAK2.
So those are the internal relays that carry the message from the receptor.
Exactly. The IFN signal comes in, but PGE2 chemically inhibits those relays from working. The message never gets delivered to the cell nucleus.
And to complete the loop, the SOX2 high TANs themselves. They must have the tools to do this conversion.
They do. They express higher levels of PDGase2, the gene for KOX2, which is the rate-limiting enzyme for making PGE2. So they're providing the enzyme needed to turn the AA payload into the final weapon. It's a perfect, self-sustaining loop of suppression.
But, I mean, that's a devastatingly elegant mechanism. But it also sounds like it opens up a massive opportunity for intervention.
A huge one.
Because we have drugs that target this pathway, specifically KOX2 inhibitors.
Absolutely. The researchers first blocked the PGE2 receptors specifically in the myeloid cells, and the anti-tumor phenotypes were completely restored in the TANs, even in the SOX2 high tumors. The conversion could happen again.
And the grand finale, the clinical payoff.
The clinical payoff. They combine the standard immunotherapy with readily available, relatively safe drugs, KOX2 inhibitors like aspirin or celecoxib.
And the result?
That combination significantly reduced tumor growth and completely blunted the relapse capacity of the SOX2 high tumors.
Wow.
It's the ultimate translation. By adding a cheap existing anti-inflammatory drug, they eliminated the molecular shield the cancer stem cells had spent so much energy creating.
So to synthesize this entire discovery for you, the listener, we have uncovered a complete axis of resistance.
It starts with SOX2 in the tumor stem cells, which acts as the master programmer. It activates the FADS1 factory,
which produces the payload, arachidonic acid, AA.
AA is then delivered to the surrounding neutrophils, where their own KOX2 enzyme converts it into the final chemical saboteur, prostaglandin E2.
And PGE2 then cuts the vital STAT1-JX signaling wires, preventing the neutrophils from converting to the anti-tumor side, which protects the stem cells and drives the cancer relapse.
It's a complete story.
This deep dive really shows that beating cancer isn't just about figuring out how to kill the tumor cells. It's about disrupting the communication networks that let the most resilient cells hide.
And targeting this SOX2 FADS1 AA PGE2 axis provides a potent new strategy. You could prevent relapse in these aggressive tSCs by simply adding a common anti-inflammatory drug to the existing immunotherapy.
Immediate clinical relevance is just crystal clear. This gives doctors a solid rationale to combine standard immunotherapy with accessible KOX2 inhibitors.
And potentially prevent relapse in patients whose tumors show the specific stem cell mechanism.
Okay, so to leave you with a provocative thought that builds on this, we focused on how PGE2 blocks the IFN signal in neutrophils. But the sources also noted that PGE2 can disrupt IL-2 signaling in T cells as well.
Which suggests the SOX2 stem cell has created a single chemical signal that achieves two forms of immune evasion at the same time.
It's running interference on multiple fronts.
Exactly. So given that PGE2 is blocking both the key T-cell activators and the key reprogramming signals for neutrophils, how do we activate long-lasting, maybe epigenetic memory in those tans? How can we make them permanently anti-tumor so they can survive high PGE2 concentrations and just keep fighting, even when the signaling wires are constantly being cut?
It's a complex ecosystem, and it seems the key to lasting cures often lies in shutting down the tumor's ability to talk to its environment. That was the deep dive. We'll see you next time.